Namaste students. We are discussing about the animal cell culture in the continuation of animal cell culture. Today we are going to discuss about the methods of animal cell culture. Which methods are used for the animal cell culture? So for the animal cell culture, we need some requirement. We are some requirement which need to be fulfilled. So what are those requirements? Before, because when we are not able to fulfill these requirements, we are not able to grow animal cells because animal cells are more complex for the growth. So they need to be cultured in more appropriate way, and they need to give some requirements which are necessary. So what are those requirements? The first one is the media. As we know that different type of cells need various requirements. Okay, they need some growth media. Okay, they need some growth factors. And when we are talking about the animal cells also need some complex requirement. Okay, they include amino acids, vitamins, salts, osmotic pressures, glucose, bicarbonate buffer system. So these are the requirements of and for the growth of animal cell culture. Similarly, the another requirement is pH. As we know that we are human and we survive at the neutral pH. We do not need any alkaline pH and the basic pH. So for the animal cell culture, the pH must be maintained near the neutrality. It may be 7.2 and 7.4. Okay, not only these factors, but also some specific requirements are also required, such as for humans, as we know that hormones are required, some growth factors are required. So these growth factors and hormones are also added to the nutrient media. Similarly, oxygen supply is also necessary. Okay, some specific media are required in the animal cell culture. So, what are the specific media in the animal cell culture? These specific media are the natural media. So, what are the natural? The blood serum is the natural media which is supplemented to the nutrient media for the animal cell culture. And for the growth, there may be found some contamination. We can find some contamination of the microorganism also. So these contamination must be avoided. For the avoidance of these unnecessary bacteria in the fungal growth, we need to supply some antibiotics. Okay, antibiotics will inhibit the growth of the microorganism. So which antibiotics can be supplied, such as penicillin, streptomycin, tetracycline, ketoconazole. So these can be supplied to the uh, media so that we can avoid the growth of bacterial and the fungal. The next requirement is temperature. As we know that humans survive at the 37 degrees centigrade, that is our basic temperature. Similarly, the cells of the animal cell culture also will survive at the same temperature, that is the 37 degrees centigrade. So for the animal cell culture, we supplement a temperature that is 37 degrees centigrade for optimum growth of the animal cells. Similarly, we can provide the l- low temperature also, up to 15 to 26 degrees centigrade can also be used. So this is the basic growth requirement. Now, what is the process to obtain the primary culture? So, as we have discussed about the what is the primary culture? Primary culture is the first cells which are grown on the culture media. Okay, so these primary cells are obtained from the fresh tissue. Okay, primary cell culture is isolate from the fresh tissue. How will isolate these cells? So that is the question. How will we isolate these cells? So the cells are isolated from the tissue, and first we need to isolate the tissue. Okay, the piece of tissue from the organs are removed very carefully, and similarly, uh, we dissociate the cells from the and the cells are dissociated enzymatically. How the enzymatically we can dissociate the cells from the uh, tissues? We can isolate the cells from the tissue by the enzymatic method, such as we can apply the trypsin enzymes. Okay, this trypsin enzyme can be used for the isolation of the particular kind of cells. Okay, these cells are then cultured. These isolated cells are then steriled using the membrane filters, and the obtained cell suspension is washed with physiological buffer to remove some enzymes. So when these impurities have been removed from the cells, these cells will be transferred to a petri dish, and they are grown. They are allowed to grow, and after growth they are subcultured for their secondary cell culture. From these secondary cell culture, the cells will adhere to each other, and after adhering they will form a line, a layer. The layer of these cells is known as the cell lines. Okay, what are the aseptic techniques are used? The aseptic techniques is the removal of and avoidance of the fungal culture and avoidance of bacterial contamination. So how we can eliminate and how we can remove them? We can remove and we can avoid these microorganisms by the aseptic techniques, such as we can use some antibiotic solutions in our media. Similarly, we can provide the sterile environment sterile environment to the process. When we will provide the sterile environment for the process, then there will be not found any kind of contamination. For example, we can use a HEPA filter for the removal of the microorganism, and we can make fumigation for the removal of the microbes which are present. So these are the aseptic techniques which are required. Next, when the culture has been conducted, we have isolated our cell lines, then what to do now? The next is cryopreservation. When we have got our cell lines, these cell lines will need to be preserved, and the method of the preservation is the cryopreservation, in which we preserve our cell in the low temperature conditions. But as we know that in the low temperature conditions, the cell can be damaged. For the remove for the problem, to escape the problem of the cell damage, we need some specific aseptic techniques. The technique is use of DMSO or glycerol. In these chemicals, these proteolytic protective agents, the cell will not get damaged in the low temperature, because we are using here minus 130 degrees centigrade. for the preservation of the cell. So this is very extreme low temperature, and at this temperature the cell will damage. So the cell may not be damaged. We use some protective agents such as TMSO or glycerol. Okay, so we provide this cryopreservation to the cells so that the cell will not go to the senescence. What is the senescence? Senescence is the uh, cell death. That is the cell death. Okay, so to minimize the chances of the cell death, we preserve our cells by the method of the cryopreservation. So these all are the methods in the animal cell culture. Our lecture is over here. Thank you very much.